Welcome back. Here we'll go ahead and start with the innate immune response. So I call it the innate immune response because I don't want you to forget that there is some sort of response when it comes to this part of the immune system. Even though this seems like, oh, it's happening every day, why do we actually really need to specify it or talk about it in any way? But it is still responding to the non-self antigens. So the innate immune response is something that universally across all human beings we will have these defenses. Here are a couple listed. So first are physical barriers. So physical barriers, think hair, skin, nails, essentially the integumentary system. Just beneath the just deep to the integumentary system, you also have connective tissues. You also have structures within the dermis and within the hypodermis. You also have structures deep to, or specialized structures deep to other really important structures, like within the nervous system. You have the subarachnoid space. And within the subarachnoid space, you might have specialized cells that will actually support the nervous system. So things like microglia, which you learned from 201. We also have deep internal structures that will present important cells if we ever need it. So things like the bone marrow or the liver, where they'll present either white blood cells or macrophages, respectively. Phagocytes, as a reminder, phagocytes eat things. So phago means to eat, cyte means cell. And we have a couple different versions of phagocytes. So we have macrophages, and macrophages are big eaters. And the way they work is they literally will go and gobble up a cell. And after they gobble up a cell, they'll either destroy their inner bits and break it down with lysozyme and other cell organelles and then recycle their products. Or they might just eat it and then blow up, essentially, and that's it. Then you've destroyed the pathogen. Don't forget about neutrophils and eosinophils and monocytes. Monocytes are macrophages. We have some macrophages that are floating around at any given time. We also have some macrophages that stay fixed. They stay in that area waiting for a pathogen to brush up against it. So think about where this might be most relevant to be in the human body. Think like things that are open to the external environment. So everything with the mucous membrane is likely going to have some amount of fixed macrophages there. We also have neutrophils. Neutrophils are one of the most abundant white blood cells that we have in the blood plasma. Excuse me, not the blood plasma, within the blood. And neutrophils are very good at being one of the first to the scene to actually identify what that pathogen is. And once it does, it eats it, gobbles it up, blows itself up, turns into pus. Very fun. We have eosinophils. Eosinophils are going to be attacking parasite-like type pathogens. And the way they work is they will release their granules, and those granules will go and latch themselves onto that parasite and either slowly disintegrate and destroy the parasite or flag it for other cells to come over and destroy it. Don't forget that the way we move these cells out of the bloodstream into the interstitial space or to the adjacent tissues is first they have to get to where they need to go. So they're going to go through chemotaxis, which means they're following some sort of chemical gradient or chemical signaling in order to get to the cell of choice. Then they finally adhese themselves onto the inside of the red blood vessel, and then they squeegee their way out, and then they go and they adhese themselves to the pathogen in question, and then they can either phagocytose it or just stay stuck to it and wait for another cell to come by and recruit its help. Next, we have immune surveillance, which everyone also has, and this is where we have those natural killer cells or NK cells. And natural killer cells essentially go around and they identify cells that are not like our cells, and they just destroy them right on the spot. The way they are able to tell if the cell is not self versus self is essentially the cells that will eat pathogens. They will eat the pathogen and they won't know what to do with it necessarily. So let's say a monocyte goes and eats a pathogen, and after it eats it, it's like, well, that probably wasn't us, but I'm not sure what it is. Let me send up a flag, and that flag will be a receptor on the cell membrane, and that receptor will be immediately recognized by a natural killer cell, and the natural killer cell will go latch on to that receptor and say, oh, okay, you have ingested something that's not, or something that's foreign, and we need to get rid of you. And so the natural killer cell will then lyse that cell or kill off that cell. This is actually how, on a side note, this is how cancer is able to dance around all of our immune systems, is by wrecking those receptors. So instead of having a cell that eats a foreign tissue send a flag and send a signal to the rest of the cells like, hey, I think this is bad news bears, I don't know what it is though, it just won't allow the cell to even raise those flags. A lot of diseases actually work like this. Okay, next is interferons. And interferons are chemical messengers that just get released from cells for a variety of reasons. Some cells might be releasing this on a regular basis, just saying, hey, I'm over here, cell A, can you come and do this? Cell B, can you come and do that? So it might just be a chemical signaling method between cells, or the cell itself could be infected with something and then send out interferons to stimulate other cells to come to the scene and do something. So it's just chemical messengers. That's all interferons are. The way we identify interferons is capital I, lowercase, that's a lie, I'm sorry. Interferons are IF. We also have something that's very similar called interleukins, IL, and we also have something similar called cytokines, which we'll talk about here shortly. And again, they're not specific. They're just floating around, triggering other cells, telling them what to do, telling them to do their normal job. Another way that we can use natural killer cells to kill off bad cells is by having them attached to the membrane. And then once they attach to the membrane, after they get that little flag saying, hey, I'm an ill cell, come and get me, the natural killer cell can secrete something called perforin. And perforin is an enzyme that literally goes and attaches itself to the membrane of the ill cell and will force the membrane to open itself into like little pores. And once the membrane opens enough pores, enough cell stuff on the inside just starts to leak out. And then the cell can't withstand that lack of structure. And then the cell just bursts and breaks apart and disintegrates. And then that's it for the pathogen. So we'll talk about perforin in another mechanism later on. So just remember perforin pokes holes. Perforin pokes holes in the membrane. Back to interferons, we have a couple different versions of interferons, alpha, beta, and gamma. Like I said, they're chemical signaling molecules. We're not going to go into the details of them in this class. You might see in like literature, if you start looking at research papers, they might specifically say like IF21, IF32, IF alpha 1F. There's just so many, and they keep getting identified all of the time. So just be familiar with interferons being chemical signalers, and that they come in a lot of different flavors. Oh, did not want to do that. Next is complement. Complement is similar to interferon and interleukin and cytokines, except a little bit different. Complement is a specific type of protein that actually floats around in the blood, and it will assist antibodies in being able to target bad cells. And complement will go and actually attach itself directly to the plasma membrane of potentially a pathogen or maybe another cell entirely. And the goal of complement, once it finally binds to that cell membrane, is to kill the cell at some point. And so there's 3 ways that complement can bind and activate this programmed cell death. And we have 3 pathways that these are called, classical, lectin, and alternative pathways. But at the final end stage, all 3 of these pathways all converge onto one single pathway, and that single pathway does something very similar to what perforin did, in that it pokes a giant hole in the membrane, and then the cell membrane bursts, and then the cell dies. So let's study each of these a little bit more closely. So we have classical and lectin and alternative pathway. What I did was I took this giant picture that you have, and I cut the top half, and then the bottom half is down here. So the top half gives you details about the classical, between the 3 pathways. Why do we have so many different pathways? Why do we care? So like I mentioned at the beginning of the chapter, there is a lot of research dedicated to the immune system, and everything is changing all of the time. And there are researchers, specifically I know of some at CSU, Colorado State, that only will study how complement works, because complement is such a diverse protein, and it has many different pathways that it can be interacting with of cells, that it can, there's a lot more to be studied with complement, essentially. We don't know enough. And when we started first learning about complement, we just started calling all of the proteins C. We were like, oh, they complement, it starts with C, we'll just start calling them C. And then they realized, well, that was dumb, because now we have all of these other proteins that are involved, and they all kind of look like complement. And so then they started naming them like C1, 2, 3, and whatever, and then they started giving them like subnames, so like C1A, C2A, C2B, and so on. And even that has become even more complicated. So we actually started understanding the complement pathway with the classical pathway, with this idea that let me make sure I get this correctly. Yeah, so with the idea that the antibodies would be floating around, and they would be tagging the antigen, they would be attaching onto antigens of the cell membrane of cells that for whatever reason needed to die. And then depending on those antibodies that were attached, we would have complement protein floating around, and complement protein would come and attach to the end, to the Y, the ends of the Y of the antibodies, as you can see in the picture. And then after it attaches, then this magical like cascade of events happen where it gets activated. And the reason why it gets activated is because that complement protein, C1 in this picture, gets divided into a couple different groups, or it gets recruited by, or it recruits other complement proteins, and then eventually finally cover this entire cell membrane with enough complement proteins that are the active form, that you finally end up going into what's called the final common pathway of the complement cascade. But then they started doing more research, and they were like, oh, wow, look, like complement does all these other things too. So then they identified these two other pathways called lectin and the alternative pathway. So lectin is a little different in that the same idea happens, except you start with lectin directly binding to the bacteria of interest, and then that lectin protein will recruit more complement proteins. And then once those complement proteins finally get split into the active form and the not active form, then you finally go through the rest of the process. The alternative pathway is different than the other two pathways in that, let me make sure I say this correctly. Is different than the other two pathways because instead of waiting for complement to actually attach to the membrane of the cell directly, it will actually become active before it touches the cell membrane. And then once that activated form directly touches the cell membrane, then we finally start this cascade of events. Okay, so do I really need to know the differences between all three of these? Not quite for this course. You do need to understand the names of the three and a very brief, like, generally what's going on between the three, and then eventually what happens when all of them converge onto the same pathway. But whenever you go into a pathophysiology course, you'll actually go into the details of what's happening during this complement pathway. Complement cascade is how I was taught it. Okay, so then let's imagine that we've went through one of those three pathways and we're finally all converging towards the end. So what happens is once we have all of these complement proteins activated, the plasma membrane forms a complex called the membrane attack complex, or MAC, M-A-C, and that allows for not only things to go in and out of the cell that will help further break apart the cell, but essentially it's a giant hole and the cell can't withstand that, so the cell lyses and breaks apart, and then the cell dies. The other option is that all that complement protein that's coating the surface can actually go and, if there's enough of it coating the surface, it can recruit other cells to come and eat that cell, saying, oh, that's a tag cell, I need to go and eat it and kill it. And we call that process opsonization, which is the middle box here. I think of opsonization as peppering the steak. So you keep peppering the steak until the steak is overwhelmingly peppered, and then it finally is, in this case, edible for the purposes of this analogy, but there's so much pepper that the other cells have to come and actually, they see it, obviously, we need to attack this cell and kill it. The other option is that nearby cells can release histamine, and then histamine can go and follow the, by chemotaxis, follow the chemical gradient to the complement protein, those activated complement proteins, and just increase inflammation to that area to bring other cells to that area to help get rid of the pathogen. Okay. All right. The last, is this the last one? I want to make sure. Oh, no. The second to last piece of the innate immune response is inflammation. And inflammation is really important because it does one of 7 things, which are all listed here. And, excuse me, that's not what, one of 7 things, sorry. But the purpose of inflammation is that you slow down the blood that's in the area that's damaged, A. B, you make it really, really hot, and by slowing down the blood, you make it really, really hot because all that heat stays in one spot. It doesn't dissipate like it normally would. And by making it really, really hot, pathogens don't like really hot environments, and so the pathogen might die. Also, by slowing down the amount of blood into that area, now you allow for the most amount of white blood cells to maybe make some sort of interaction with the pathogen that might be in that area. So imagine that you get pricked with, like the picture on the left, you get pricked with a needle that has some sort of pathogen on it, and then that pathogen comes off the needle, goes into the tissue, and the surrounding cells are like, uh-oh, that's bad news bears. I don't know what that is, but that's definitely not us. And so the mast cells that are surrounding that area will automatically release histamine. Histamine is going to be in these granules that are within the mast cell, and that histamine will trigger the immediate surrounding blood vessels to get red, to dilate, to swell up, to allow all of that heat to come to that area, and it might even be a little bit painful. The reason for the painful signal is so that you recognize that something is happening in that area, which sounds kind of like dumb, right? But like, how many times have you like scratched your hand and then looked at it like a week later and you're like, oh, I wonder what that's from. I don't remember that, right? So the pain is supposed to be there to help remind you, like, hey, something's happening here. Don't mess with this area, or help treat it if you need to. Make sure it's clean, or protect it from being damaged by something else later again. Once you get all that histamine to that area, now you've got an increased amount of blood moving to that area, which means you have more white blood cells to that area. And then the idea is that once you have more of those white blood cells, they can go and clean up the damage as they need to. You also have a release of cytokines. Like I said, cytokines are going to be another example of some chemical messengers that cells can release, and cytokines are really important in helping recruit other cells to the area. And then finally, once the pathogen has been taken care of, the tissue has been recreated or scarred or however it needs to heal, then you finally have entered that tissue repair stage. Okay. Last but not least, we have fever. Fever is really important in being an innate immune response for us because, like I said, pathogens typically don't like staying in a really warm environment. They don't do well. They can't replicate. They can't do their regular functions as they normally would. The reason why pathogens stick around in our bodies is because our environment, like our physiological environment, is really rich in nutrients. It's really warm. It's really wet. It's which sounds gross, but that's true, and it's a perfect space for the some pathogens to replicate themselves, to hang out and hide in other tissues, and in order to prevent that, we might initiate a fever to help decrease the ability of the pathogen to actually live in that environment. I'll see you in the next one.